You may be curious, what's the difference between gene therapy, cell therapy, and gene editing? And why are they usually mentioned together? Well, all three are fields of research with similar goals in mind. Treat disease by changing the blueprints to our bodies at a microscopic level. Our genetic makeup. But how do they do it? That's where the differences lie. First, let's talk gene therapy. It's the introduction, removal, or change in genetic material, specifically DNA or its molecule cousin RNA, in the cells of a patient to treat an inherited or developed disease. There are a couple ways gene therapy can be administered. Typically, genetic material, such as a working copy of a gene, is transferred into the cell. Once in the cell, the transferred genetic material changes how proteins are created, so they can do the work for the gene that's not functioning. Proteins are the building blocks of how we function, so they make a big impact on our health. A vector, which is often a virus, delivers the gene into the cell. Don't worry, the viral genes that could cause disease are removed. Then the vectors are directly injected into the patient's body. The new working gene is able to alter the behavior of the protein within the cell, whereas cell therapy uses living and sometimes genetically modified cells to help treat a disease. These transferred cells can be from the patient or someone else. Cell therapy is routinely administered through blood transfusions. Genetically modified cell therapy is when a cell is removed from the body and the gene inside is altered, and then it is put back in the body. This combines components of gene and cell therapy approaches. For more on that, here's Dr. Rain Rouse.
An example of this approach is CAR T-cell therapy. We take an immune system cell, usually a T cell, and we introduce a new gene into the cell carrying a specialized receptor that's been trained to only target molecules that are on the surface of cancer cells. Large numbers of these changed cells are grown in the laboratory and then returned to the patient's body so that they can kill cancer cells.
For many years, gene therapy has been a hope on a distant horizon. But today, with the first gene therapies approved in the US and others looking promising in research, people affected by genetic diseases are beginning to see gene therapy as a real possibility in their lifetime. Can the changes gene therapy causes be passed on to future generations? There are two different types of cells, somatic and germ. Germ cells exist as eggs and sperm and only have one set of chromosomes. These are the cells that are passed to offspring. Somatic cells are present everywhere else in the body and have two sets of chromosomes. Right now, gene therapy is only given to somatic cells, and therefore the changes it causes cannot be passed on to offspring. It is possible that in the future, gene therapy could occur in germ cells, but right now that is not happening. How long is the treatment? What does it entail? And does it reach every cell in the body? Treatment can be given systemically or injected into the affected area. Systemic treatments are given via IV and are typically longer, up to a few hours. This also depends on your weight. A large adult will receive more gene therapy than a small child, so the treatment will be longer. Injected treatments are completed immediately, as soon as the fluid is injected into the area. Gene therapy that is administered systemically does have the potential of reaching all somatic cells, depending on the dose, but gene therapy is more often targeted to only the diseased cells. Why are there variations in responses? There are many reasons for this. One is that every individual has different DNA, so gene therapy may work better for some than for others. In addition, there is variability in the success of the drug, how effectively the gene is delivered to cells, how many cells need the gene to function, and how much enzyme the introduced gene produces. Is it safe? What are possible side effects? As with any drugs, side effects are possible and expected. Normal immune system responses such as fever are common, but severe responses do occur. But it is important to remember that no drug can be given to humans until there have been preclinical safety studies and the data approved by the FDA. Will it be only those with severe or life-threatening disease? Before gene therapy can become widely available, it will likely be tested on more severely affected patients. Most scientists are currently focusing on development of gene therapies for severe disorders. In addition, individuals who have already been exposed to the virus in which the gene therapy is packaged will not be eligible for treatment. The viruses used in gene therapy are naturally occurring and benign, but still foreign to the body. Because of this, individuals can come in contact with the virus and antibodies develop. Those antibodies would destroy the gene therapy before it could be effective if that person were to be treated with gene therapy.